Hello. In this section, I will explain almost a detailed explanation on the essential dogma and protein structures. I do hope you will be able to follow through, and only advice I have for you is to continue finding extra information about this topic. It will certainly help to brush up your knowledge about essential dogma and protein structures. Let's get ready and begin. So, the essential dogma of biology describes just that. It provides a basic framework for how genetic information flows from the DNA sequence to a protein product inside cells. This process of genetic information flowing from DNA to RNA to protein is called gene expression. DNA sequence information is converted to RNA in a process called transcription, which is the first step of gene expression, where a segment of DNA is copied into RNA polymerase. First, the double-stranded DNA molecule is partially unzipped, and an enzyme called RNA polymerase literally copies the gene's nucleotides one by one into an RNA molecule. Like DNA, RNA is made of a particular sequence of nucleotides. Unlike DNA, RNA has only a single strand and is a more fragile and temporary molecule inside the cell. Very importantly, RNA is small and can easily exit the nucleus and go to the cytoplasm, where proteins are made. The sequence encoded in the RNA molecule is decoded and converted into an amino acid sequence in a process called translation. Transcription is a process by which the information contained in a section of DNA is replicated in the form of a newly assembled piece of messenger RNA, mRNA. Enzymes facilitating the process include RNA polymerase and transcription factors. In eukaryotic cells, the primary transcript is pre-mRNA, messenger RNA. Pre-messenger RNA must be processed for translation to proceed. Processing includes the addition of 5-cap and a poly-A tail to the pre-mRNA chain, following by splicing. Alternative splicing occurs when appropriate, increasing the diversity of the proteins that any single messenger RNA can produce. The product of the entire transcription process that began with the production of the pre-messenger RNA chain is a mature messenger RNA chain. The process of translation can be divided into the stages of initiation, elongation, and termination. Initiation involves at least 3 other proteins called initiation factors to help the messenger RNA bind to the smallest subunit of the 2-unit ribosome. It is bound to the correct location using the initial codon AUG on the messenger RNA. The next phase is elongation, or adding other amino acids to the building polypeptide chain. According to CARP, a step of elongation can be accomplished in about 1 to 20 seconds, so a small protein of 400 units could be assembled in about 20 seconds. It is suggested that most of this time is used sampling the amino acid carrying transfer RNAs in the cytosol to find the ones for which there is a codon-anticodon match for the next codon along with the messenger RNA pattern. The messenger RNA does not contain all the information for specifying the nature of the mature protein. The nascent polypeptide chain released from the ribosome commonly requires additional processing before the final product emerges. For one thing, the correct folding process is complex and vitally important. For most proteins, it requires other chaperone proteins to control the form of the product. Some proteins then excise internal segments from their own peptide chains, splicing the free ends that border the gap. In such processes, the inside discarded sections are called enteins. Other proteins must be split into multiple sections without splicing. Some polypeptide chains need to be cross-linked, and others must be attached to cofactors such as the heme before they become functional. On the screen right now, you'll see you are looking at all 4 different types of protein structures. These types of structures are essential information about protein functions after they were translated during the translation process, as explained. Protein structures range in size from 10 to several thousand amino acids. By physical size, proteins are classified as nanoparticles between 1 and 100 nanometers. Very large aggregates can be formed from protein subunits. For example, many thousands of actin molecules assemble into a microfilament. A protein may undergo reversible structure changes in performing its biological function. So let's summarize the topic that we have learned today. In this topic, we have learned about the concept of central dogma and 4 types of protein structures in general.